Transcribe. All right, just doing an audio test here. Audio test. Why is it not showing up? There we go. Okay. All right, cell bio. Hope everyone's doing well. So the other day when we had the lecture on, that was lecture 6 on September 24th, we covered sections 7.1, 7.2, and 7.3. And I forgot to put the microphone on for section 7.3, so I'm adding a quick supplementary lecture so you guys have a recording that you can review. So this should be pretty quick. So this chapter I'm not really stressing too much, but I do want you to know the concepts, just not really the mechanisms and the specific examples. Epigenetics is hugely important. It's really important. And that's why we need to definitely cover the concepts. But the mechanisms are, they're kind of new. And, there's just only so many hours in the day. So concepts over mechanisms for this chapter. So when you're studying, be sure you're looking at the PowerPoint slides as like a study guide on what you're responsible for. Okay. All right. So like I said, this chapter is on chromatin and epigenetics. These are really fundamental to understanding how cells regulate gene expression beyond the DNA sequence itself. Right? Chromatin structure, chemical modifications, they can either promote or restrict access to the genetic code by RNA polymerase. Right? So these epigenetic mechanisms provide a way for cells to also remember transcriptional states, allowing cells, allowing cell identity, and its function to be maintained even after cell division. And that's why it's a form of inheritance. Okay? All right, so, whoops. All right, so so what is being inherited are the modifications to the proteins that package DNA. Okay, those are the histones, right? So the histones are involved in wrapping, packaging, and tightening the DNA. And because of that, DNA can kind of exist in different states of compaction. And that directly influences transcriptional activity. Heterochromatin is tightly packed, limiting accessibility of the transcriptional machinery and leading to gene silencing. In contrast, euchromatin is loosely packed, allowing transcription factors and RNA polymerase to access promoters more easily. This distinction is a central concept in epigenetics, since changes in chromatin condensation can switch genes on or off without altering the DNA sequence itself. Okay, remember in class, to help you kind of remember this, think hetero, hard, compact, unable for RNA polymerase to access because it's so packed down and hard DNA and protein. All right, so here's a picture of the decondensed chromatin. So it's important terminology, decondensed and condensed, right? So, all this stuff is still packaged, right? You've got nucleosomes here. It's just tighter in a condensed chromatin model, right? So, you can see the nucleosomes here. You remember the histones are, it's an octamer. You have the core histone. And then it also has these flexible amino terminal tails that extend outside the nucleosome. You can see that here, like little wiggly things that are coming out. These tails are accessible to enzymes that can add or remove chemical groups, such as acetyl or methyl groups. Because histones are positively charged and DNA is negatively charged, these chemical modifications influence how tightly DNA is wrapped. This makes histone tails crucial points of regulation, functioning almost like molecular switches for chromatin assembly. All right, so histone acetylation is strongly associated with transcriptional activation. Enzymes called histone acetyltransferases, or HATs, will add acetyl groups to the lysine residues on the histone tails. This neutralizes their positive charge, and as a result, histone DNA interactions loosen. It makes the promoters more accessible to transcriptional machinery. All right. On the other hand, conversely, histone deacetylases, or HDACs, remove these groups, restoring the positive charge, which tightens DNA packaging and represses transcription. Right? So it's the combination of HATs and HDACs, right, that do this stuff. But all that modification is occurring on these histone tails, right? So, together, HATs and HDACs act as co-activators and co-repressors, dynamically regulating gene activity depending on whatever the cell needs. Co-activators, co-repressors, HATs, HDACs. Modification is occurring at the lysine residues. All right, so here's the amino terminal tail domain, right? This is the histone folded domain. So this is what's folding up and creating the components that make up the octamer. See the lysine residues here. Lysine is readily acetylated by acetyltransferases. It's also readily deacetylated by HDACs. Okay, you can see it acting as a co-activator, right? Because it's acetylating, and as a co-repressor because it's deacetylating. All right. Histone modifications, patterns of histone modification, right? So acetylation means it's transcriptionally active. Methylation is another modification that can occur to the histone tails, and that usually results in the repression of transcription, and also phosphorylation. This can result in chromosome condensation, and it's usually a result of DNA damage. Okay. All right, so here is an example of the histone tail. You can see multiple sites. They can be either methylated, acetylated, or phosphorylated. And it's a dynamic combination of these things that will ultimately dictate how active it is. Methylation, however, is almost exclusively inactivating chromatin, right? Because if it's methylated, it's basically blocked. So RNA polymerase cannot bind to the DNA because it's can't bind to the DNA. All right, let's see here. All right, so there are also chromatin remodeling factors, right? So this goes beyond chemical modification. Cells can use ATP-dependent chromatin remodeling complexes to move or restructure nucleosomes. These remodeling factors can slide histone octamers along the DNA, evict histones from promoter regions, or even exchange histone variants. This repositioning increases accessibility of promoters to general transcription factors and RNA polymerase II. Very importantly, remodeling factors are often recruited by transcriptional activators. This creates a very coordinated system where DNA binding proteins direct local chromatin restructuring to promote or repress gene expression. Right, so you can see the chromatin remodeling factors, right? And so here you can see the nucleosome is basically being displaced, right? So in cohort with an activator protein, it's displacing it, also recruiting mediator RNA pol II to the DNA. All right, histone modifications and epigenetic inheritance. So one really remarkable property of histone modifications is their ability to reinforce themselves, right? So modified histones recruit enzymes that propagate the same modifications to nearby histones, creating very stable chromatin domains. This is this process is the basis of epigenetic inheritance because the ability to transmit patterns of gene activity from one generation of cells to the next without altering the DNA sequence, that's what we're talking about here. Repressed chromatin can remain repressed after DNA replication. That's because the histone, the modified histones are distributed to the daughter cells, and they guide modifications of new histones without altering the DNA sequence. All right, so basically, incorporation of new nucleosomes can instigate, the recruitment of even more nucleosomes. All right, let's talk briefly about DNA methylation. So DNA methylation is a very powerful silencing mechanism. When promoters are heavily methylated, gene expression is repressed, and this state can persist through multiple generations of cells. Methylation is also reversible. So demethylases, also DNA repair mechanisms, can restore cytosines to an unmethylated state, and that can reactivate silenced genes. So it's a reversible control. But it's typically that it occurs at these CPG, where they're called CPG islands. It's just like CG repeating over and over again. And also at if they occur at promoter regions, you definitely get transcriptional repression. Okay. We talked about during DNA replication, right, in E. coli. So methylation is a event that occurs after DNA is replicated, but it is it's a, I don't want to say permanent, but it's a very solid form of modification to the DNA, and that's why it can be inherited. All right, there's DNA being methylated. Cytosine gets a methyl group on that 5' carbon and becomes now 5-methyl cytosine, which is a larger, bulkier nucleotide that can inhibit DNA polymerase from traveling down the DNA. All right, genomic imprinting. So there's an example here of the gene H19. Genomic imprinting is a good example of how DNA methylation controls gene expression. So in this specific example, in genomic imprinting, only one allele of a gene is expressed depending on the parental origin. For example, the H19 gene is methylated and silenced in male germ cells, but remains unmethylated and active in female germ cells. So after fertilization, the paternal allele is silenced while the maternal allele is expressed. What does this do? Well, this ensures that the parent of origin specific expression and demonstrates how epigenetic marks can dictate inheritance patterns, again, independent of the DNA sequence. See the genomic imprinting there, right? Egg, sperm. There's the H19 gene, and it is methylated in the father, right? So all you're going to get is expression of that allele from the mom. It's like a form of gene dosing. All right. So, yeah, that was it. Short chapter, but I just want to make sure you guys had something to, to go on. There's a lot more to epigenetics. It's a huge field. And really, it'd probably be best to do like a topics course on something like that. But that's the basis of it. All right, so I will see you guys for the next lecture. And we're getting into some tough stuff here. We're getting into translation Translation is a very, very intricate, detailed process. There's a lot of control of it. There's a lot of proteins involved in it. There are proteins involved in the initiation and the elongation. So we're going to learn it at a much detailed level than you've probably learned it before in Bio 1. Okay. All right. I will get this posted to Canvas, and I will see you guys soon.